So my name is Samir Parekh. I'm a physician scientist at Mount Sinai in New York. I treat patients in the clinic as well as have a research interest, both lymphoma and multiple myeloma. And my lab has been continuously funded by the Chemotherapy Foundation for almost 15 years now. I'm very thankful to all the patients and donors that support the foundation. My lab has actually focused on cancer called multiple myeloma. And in fact, my main role at Mount Sinai is being the Director of Translational Research in multiple myeloma. And myeloma is a disease of plasma cells. In this disease, for a variety of reasons, plasma cells, which we all have to make antibodies, start growing in an uncontrollable manner in certain individuals. And the growth of these plasma cells leads to the production of an abnormal protein called the M protein, which is detected in the blood. Most of us go to our GPs for annual checkups. Thankfully, when these annual checkups are done, this abnormal protein can be picked up because the checkups involve some blood tests. And we are picking up this disease earlier and earlier in most instances. 10 years ago or 20 years ago, we were seeing patients that would come in with very advanced stage of the disease, with fractures, with kidney failure, and those presentations, thankfully, have become much rarer now. We are seeing patients mostly being referred by other doctors because they've come in with this high protein in the blood, and then we are able to counsel them with effective therapies that are now available and work in more than 90% of the time are given outpatient to reduce the burden of the disease and put patients into remission very effectively. The problem in this disease, however, is not the initial portion. Most patients get into remission quite easily. It's when the disease comes back. This can happen a few years later or even decades later, and that's when we have to change therapies to second generation or third generation of drugs. And thankfully, when I was in training, there were only 2 drugs available, and the median survival for most patients was just 2 to 3 years. Now, we have a panoply of drugs. We have over a dozen approved drugs in myeloma, with several of the drug approvals just occurring in the last 2 to 3 years alone. And in fact, the median survival is now exceeding 8 years and beyond, and a fifth of the patients have more than 10 years median survival. At Mount Sinai particularly, we've treated over 200 patients with the latest immunotherapy class of drugs called CAR-T and bispecific antibodies. And I can tell you, in my own clinic, we have patients living for 3, 4, even 5 years out with this class of drugs after failing all the conventional immunotherapies and FDA-approved options before. So there's a lot of hope with these new treatments. And what we are trying to do now in research is to tackle the class of patients that are not benefiting either upfront, that have challenging disease and relapse quickly, or the class of patients that are relapsing despite these new immunotherapies. And the way we are going about doing this is by understanding both the tumor and the microenvironment around the tumor in the bone marrow in great detail. And again, thanks to Chemo Foundation and thanks to other research support that we've had, we have been able to do this at very high resolution with the new technologies available at single cell level to really understand what is going on. And with the help of this new information, try and preempt disease before it actually relapses. So I'm going to show you a couple of examples of how we are doing this. One way we are doing this is by actually using very advanced computational methods. We have a large computational group here, which has published several papers where, just like in an orchestra, we have different instruments. Earlier, we were only able to look at one aspect of the tumor at a time. Now, with very powerful computational techniques and supercomputers, we are actually able to integrate far more layers of information and develop classification systems that are multimodal. And in fact, we published a couple of years ago the first integrated system, classification system for myeloma. And this has helped us to really understand and classify the disease into far more classes that have impact in choosing even treatments for patients that are relapsing after CAR T. This is one such example of patient that was relapsing after CAR T and actually was destined to go to hospice. And instead of going to hospice, we were able to treat him with a drug based on his molecular signature. And the drug worked. In fact, instead of going to hospice, he went into remission and he's still alive, and he's actually doing well more than a year and a half of after his disease had relapsed. So this has been published, and now we have an NIH grant to actually expand this kind of approach to other patients. So a clinical trial is about to get underway to allow us to do this with a large number of patients, and I'm very excited to do this. My colleague, Dr. Keyboard, is actually pursuing a project that is very important, and especially, again, as part of myeloma awareness. I hope that you'll find this interesting. His project involved looking at a large number of patients. He looked at over 600 patients and then reproduced this in several thousand patient data sets to find that about 6% of patients have an inherent tendency to develop myeloma that can be carried on in the family. So we have always thought of myeloma as a sporadic disease, and never really counseled our patients to think about it as a familial disease. But looking at the genetics now may be important, as a small subset, about 5 to 6% of patients may indeed carry genetic mutations that make them predisposed not just to myeloma but other cancers as well. These cancer syndromes, as they are called, include genes like BRCA1 and 2, which can predispose to breast cancer and other cancers. And it's really important to pick these up. As myeloma becomes more treatable, we want our patients to continue surveillance, and this may have impact not just to the patient but also to their children and other relatives. So we are setting up a completely different pathway in the clinic, which involves genetic counselors and other collaborators, where these patients can be picked up and counseled appropriately. So this is an important finding for a small group of patients, but young patients, patients that have a family history of other hematological malignancies or prior history of other cancers, particularly need to be aware that they may need genetic counseling. And this is an important step that should be under consideration even as their myeloma treatment may be ongoing. And as they live longer, this is something to keep in mind that they should consider doing electively and discuss with their doctors. Coming back to the exciting treatments that I was talking about, CAR T and bispecifics are the two exciting classes of treatments that have been approved for myeloma. At Mount Sinai, we have been using these for several years and are seeing patients benefit from these every day. I think that these are really revolutionary, but they are not perfect. We are seeing that patients can still relapse after getting these treatments. And our main job now as physician scientists and as researchers is to figure out how we can help the patients even after the relapse. And one of the main things we've done is we have tried to figure out not just treatments, but how we can better manage the patients while they are getting these therapies, better manage the side effects, and how to sequence these therapies. So one of the side effects that we actually published on was on a patient who developed a very rare side effect. This was truly remarkable. He was actually a musician who had gotten CAR T and went into remission. And a few months later, he came to clinic and had a very unusual symptom. He wasn't able to hold his guitar and practice. And we went deeply into his case and found that he was developing a very rapidly progressive case of Parkinsonism. When we really investigated was because of the CAR T actually expanding, and we think it actually went to a part of his brain that controls motor function. This is a very rare and unusual event, but there have been a handful of patients like this now across other CAR T trials and with approved CAR T as well. So it's something that we described, which is now out there and in fact is gaining recognition. So people are aware of it, physicians are aware of it, and there have been mitigation strategies that have been developed to watch for this side effect and reduce this side effect. We are using high resolution platforms to also look at complications. And the last few years, all of you all know we've gone through the COVID pandemic. We actually use research that we had developed for myeloma and for understanding the immune system in myeloma to then pivot to COVID research. And we had several publications during the COVID pandemic to understand how myeloma patients can not mount the immune response similar to healthy individuals. And we actually found myeloma patients were actually more vulnerable to COVID. And even after they were vaccinated, certain subgroups of myeloma patients, because of both the disease and certain treatments like daratumumab or BCMA targeting treatments, remained vulnerable. So we recommended that these particular patients really keep an eye out for COVID and remain masked and make sure that they are properly vaccinated, not just the patients, but their family members, their relatives, really make a protective cocoon of vaccinated individuals all around themselves. And this is the message that we've been trying to give out, and I hope this is a part of myeloma awareness as well. Even though COVID is now less frequent in US and New York, it is still something that can come about in waves, as we have seen over the last few years. But it has helped us understand the immune system a lot more. And I think now we are going to pivot back by taking some of these findings and learning more about myeloma as a result of our research into COVID. One of the things, for example, is that COVID was associated with cytokine release syndrome. It turns out that the new immunotherapies can also cause cytokine release syndrome, and we are studying both of these using very advanced technology. There's a lot of crosstalk between experts here in immunology. In fact, the floor that I'm sitting on is actually an immunology floor where, thanks to the location, we constantly get exposed to, and there's a lot of crosstalk with T cell biologists and B cell biologists. And this is an example of a paper that we wrote with some excellent immunologists who did a lot of COVID research based on the myeloma research we had done. And in collaboration with them, we had actually had a publication with the immunologists and are doing some additional research in CAR T patients, where currently CAR T patients need to be admitted for several weeks after getting CAR T because of the cytokine release syndrome that I mentioned. To make this easier for them, we would love to have this period be decreased and give the CAR T more as an outpatient treatment. To do this, one of our fellows, Dr. Rajiv, has actually gotten an ASCO award to use some new technologies like wearable monitoring. With the help of this, she has shown that we can actually predict when CRS is going to happen up to an hour or an hour and a half before it can actually happen and get picked up by standard of care measures. And this is going to be presented at the ASCO meeting later this year. But we are planning to make an early warning system, very much like the weatherman gives you an early warning system for a storm. We want to predict the cytokine storm before it happens with the help of this research and make it more predictable. And hopefully one day we can give CAR T and bispecific therapy as outpatient. And last few minutes, I'm going to talk about how we are studying CAR Ts to make them better and how to make bispecific therapy better. we are using these research tools and working with immunologists. So Dr. Burakov was actually the immunologist and head of the cancer center that recruited me to Mount Sinai. He has actually found a new drug against a gene called HPK1. This is a very important gene that regulates T cell function, and he has developed different inhibitors for it. This is able to then actually rev up the T cells again, and I think this has a lot of promise in actually reinvigorating the CAR Ts and bispecifics. And this is the kind of collaboration that I see has a lot of promise. Another collaboration that we have is with BMS. They have new types of immunomodulatory drugs. You all may be all familiar with Revlimid or pomalidomide. These are approved thalidomide analogs. There are still next generation drugs. Iberdomide is one that is soon going to be approved, probably in the next year or so. And this is going to be even capable not just of killing myeloma cells, but activating T cells and NK cells. This is research we did with Oxford University that is actually showing that the drug may be helpful both directly and indirectly. And we have actually now shown that it can even help CAR Ts. So we are working with Emory University in an investigator-initiated study where we are combining it with another antibody against SLAMF7 called elotuzumab, and the study is underway. The first few patients have already been treated and are responding. This is the kind of research where I think we can really take CAR T and bispecifics to the next level to improve the treatments even more. Another interesting link before I wind up is with the endocrinologists at Sinai. So Dr. Emily Gallagher is one of our endocrinologists. She has found a link between obesity and diabetes and myeloma. And in fact, using a very elegant mouse model that she developed, she showed that when there's a diabetes or pre-diabetic state, the growth of myeloma is much more. So we can not only treat myeloma by giving chemotherapy, but even controlling the metabolic environment may be very important. So there are pathways that can stimulate the proliferation of myeloma cells by virtue of the metabolic environment. And in fact, thanks to researchers over here that make drugs against certain pathways like Dr. Prem Kumar Reddy, we've actually been funded to do a clinical trial with his compound. This is a drug, it had a number before, now it has a name called narazaciclib. And we are working with the company making this drug. And thanks to some funding, we are actually launching a clinical trial this year for relapsed myeloma patients. So with that, just say thanks to all the other physicians in my group and the lab. And especially thanks to the Chemo Foundation for all their support over the years. A lot of gratitude to the patients that have been part of this research and contributed generously their samples and time. If you all have questions, please reach out. You can find my email. We have a web page, and you can even go through the Chemo Foundation and contact us, and we'd be happy to respond to any questions that you all have.